Hello everybody and welcome back to the Sanctuary. I'm your host, Professor C, and we'll be looking at some more A&P today. Specifically, we're going to check out what's going on at the neuromuscular junction, where a neuron talks to a muscle and tells it what to do. Let's check it out. Okay, so the question I'm asking is, what is the common link between a black widow spider, a debilitating muscle disorder, a debilitating brain and body disorder, and up here at the top right, a toxic nerve agent known as sarin? Well, it says three letters, A, C, H, a neurotransmitter called acetylcholine. So let's discuss this chemical more and why it's so important in physiology of the nervous system and muscles. So our last talk ended with a discussion of excitation of a neuron. And remember, we had plus on the outside and minus on the inside, and we hit this little area of the membrane with a sufficient stimulus to wake up the neuron, is what we said, and generate an action potential. And then, of course, this depolarizing wave set off a domino effect of propagation and sent action potentials zipping down toward the terminal end of the axon, or what's sometimes called the terminal knob, or the terminal bouton, or the terminal button sometimes. There's a lot of different terms for it. I'm just going to call it the knob right now. And so if you want to hear those details, check out the talk that I just did before this. But at some point, these action potentials are going to run into the end of the line here. They're going to run into the terminal knob. So waiting over here, and we'll just keep this real simple. We'll just say a muscle fiber is waiting over here. Remember, a muscle fiber is a muscle cell, and inevitably we want to contract that fiber. That's what we're trying to do in these series of excitation and contraction talks. But we have a gap here, right? And we have to get across that gap. We're not going to shoot a lightning bolt across there, so we have to do something else. And it involves this chemical called acetylcholine, also known as ACH. So the statements that are below are reinforcing this picture I've just drawn. It says the terminal knobs of a motor neuron, and remember, this over here is a motor neuron. We can go ahead and remind ourselves of that. I'll just put motor N right there. And the muscle fiber, which I've already labeled, is separated by a space. Now, right now I've just called it the gap, but technically it's called a neuromuscular junction. You can argue that it is a form of a synapse, but usually we talk about a synapse being between neuron and neuron. So I'm going to use the term neuromuscular junction, or sometimes I'll just write NMJ if I need to describe this gap. Now, it's not a very big gap. It looks on most figures like these two structures are actually touching, but there is a space in between them. So what we're going to do, instead of sending a lightning bolt across or having these action potentials jump across, is we need to know that inside of this terminal knob and other terminal knobs that support other fibers are vesicles. And they're going to be loaded with ACH. I'll just put A in here right now because I don't really have enough room to write the ACH. But those are going to be loaded with acetylcholine. And that's what it says right here. Stored within the terminal knobs is a neurotransmitter, sometimes just labeled NT, called ACH. And that's usually what it's called when you hear it in talks and lectures, is ACH instead of acetylcholine. But it's the same thing. Now, where do we get acetylcholine from? Well, it's just going to remind us that most things in the body happen through some sort of enzymatic reaction. So there's an enzyme named CHAT, choline acetyltransferase, and it takes acetyl-CoA that's in the terminal knob and choline that's in the terminal knob and fuses those together and create ACH, acetylcholine. So what are the events? How do we actually get this information from one side to the other, from the neuron to the muscle fiber? Well, it says, I'll just kind of make it bigger here. There's our giant terminal knob here. And here are our action potentials zipping, propagating toward the terminal knob. So it says here when nerve impulses, and that's another way you can discuss an action potential, is to call it a nerve impulse, if it is actually on a nerve, reach the knob. When they slam into the knob, the permeability to calcium greatly increases. So what we're going to see is an influx of calcium. Calcium has 2+ charges. Sometimes you can write it like this, Ca2+, or you can write it like I've done in my picture, Ca++, where it kind of looks like it says cat. So that is calcium coming in. Now this calcium will activate a protein here. And you see it in pink right there, synaptotagmin. It's not just one protein. There's a whole class of these proteins called synaptotagmins. And so I'm just going to put SYN instead of writing out that entire crazy word. Synaptotagmins are turned on. And what this does is take acetylcholine, which is, normally just stored here in the terminal knob, and it puts it into vesicles. That's the proteins that will help vesiculate acetylcholine. Now, what are we going to do with a vesicle when it's here in the knob? Well, remember, we have to get over to the muscle over here. I'll just put M for muscle. We need to get this chemical out. So what we have to do is some secretion. So what happens next? Well, we're going back to the terminal knob here. Remember, we had an action potential zip down, bang into the knob. We had calcium come in. This stimulated or activated proteins called synaptotagmins. And those synaptotagmins helped us vesiculate this acetylcholine. And now the acetylcholine is going to be exocytosed into this neuromuscular junction. Remember, waiting over here is a muscle fiber, and it will have receptors, special nicotinic receptors, designed intentionally to bind to ACH when it gets there. So we're going to release the ACH into this gap. And you might be asking yourself, if we're just going to move across here through some kind of diffusion, what is stopping it from leaving the gap? And the answer is nothing is stopping it. Some of the acetylcholine actually does leave the gap when it is secreted, but we'll get to that in a moment. So we're going to be released through exocytosis and diffuse across the neuromuscular junction. What we have to keep in mind is that diffusion is slow and random process. And you might think that's a strange type of event to rely on something that is speedy like neurotransmission. But we get around this slow and random process very simply. And let me draw that right here. Here's a terminal knob, and here's a muscle fiber waiting for the signal. So in reality, that's very, very close, right? There is a gap, but it's a very narrow gap. So we can get over the slow part simply by making the neuromuscular junction very, very small. We can get over the random part simply by thinking about, say, sea turtles being born on a beach. When the baby sea turtles are born, there's a lot of them, and they all start running toward the beach, but not all of them make it, right? There are more sea turtles that are born than need to be born to support the next generation. So, yes, some of the sea turtles will wander off. Some of them will be destroyed by enzymes inside the gap we'll talk about in a minute. But some of them, enough of them, should make it over finally to the harbor. It's almost like boats randomly banging into each other until they find their place in the harbor. And when they're bound with those ACH receptors on the muscle fiber, the next part of the story occurs. Okay, so I mentioned just a moment ago an enzyme that could eat the baby sea turtles, right, as they're moving across the neuromuscular junction. A better way to say that, there's an enzyme that can terminate the effects of acetylcholine by simply ripping it into pieces. And its name is acetylcholinesterase, another mouthful. Usually we can shorthand that by just saying ACHE or something like that. So we don't want the muscle to be stimulated and then stimulated and then stimulated by all that acetylcholine that we've dumped into the gap. So we need to terminate it by breaking it apart. And so if here is an acetylcholine, I'll just put an A here, and there is the enzyme ACHE. What it does is it takes that acetylcholine and it breaks it into acetate and choline right here. And then the acetate and choline, I can just draw a terminal knob here, even though it's on the wrong side, as our other drawings. It will be, the choline and acetate will be reabsorbed or reuptaken back into the terminal knob, where it can be turned back into acetylcholine. Okay, at the end of the story, we have what's called an end plate and an end plate potential. So let me change my ink color. So here's what's going to happen. Remember, we had action potentials zipping toward the knob. This stimulated calcium to enter the cell. This stimulated synaptotagments, which I'll just write like that, to help us vesiculate ACH, which is the little red balls, into vesicles. We will exocytose them, or secrete them, from the terminal knob into the neuromuscular junction, and you can see those being dumped out there in abundance. But what happens when it does bind is it will open ligand-gated channels on the sarcolemma, which is the muscle fibers membrane. So a ligand-gated channel is something we haven't really talked about in here, but it depends upon a specific chemical binding to it to turn it on. In this case, the ligand will be ACH, and when it binds to the ligand-gated or ligand-gated channel, this will allow depolarization to occur. It will open sodium gates and so on, and this will cause action potentials to move up toward other voltage-gated channels. Remember, we see that ball and chain that we saw in the previous lectures, and it will do the full-blown flood of sodium in, causes a huge depolarizing wave, and then we get this series of action potentials that will propagate along the surface of the sarcolemma. Now remember though, we don't want action potentials on the surface. We want it to get the action potentials deep into the muscle where the sarcomeres are, so we can contract them and make the fiber shorten. So thanks for watching that one. That was pretty complicated, but check out some other videos in this series if you want to learn more about A and P, and I'll catch you on the next one. Bye-bye.